one life science and medicine winner discovered a blood gene, which paved the way for a genome-editing therapy of sickle cell disease and beta thalassemia, which are blood diseases that affect millions of people worldwide. In Bethesda, Maryland, in the United States, Swee Lay Thein is a hematologist and a medical doctor. She leads the Sickle Cell Branch at the U.S. government's National Institutes of Health.
I'm really very much a physician-scientist. My research is patient-driven. I see the patients and I want to address questions to try to help find new therapies.
Today, she is seeing one of her patients, 61-year-old Yao Loco from Ghana. He has the most severe form of sickle cell disease, a genetic blood disorder.
And it causes a very, very painful crisis. It's very, very painful. And mine used to always start from my hips and then my chest and my knees.
He avoids strenuous activities such as playing football.
You exert yourself so much, and then that is how you go into this mini crisis. So it affected me, and then I was in the hospital most of the time. So it affects you. You get depressed sometimes. You get stressed out.
He would go to the hospital as often as once a month to treat a pain crisis. People get sickle cell disease from inheriting one mutated gene from each of their parents. Hemoglobin is a protein in the red blood cells that is responsible for delivering oxygen to the tissues. The mutations affect the production of hemoglobin. The red cells turn into a sickle shape and stiffen. It cannot flow through the blood vessels properly, stopping blood supply to the tissues, causing pain crises. Swee Lay Thein discovered the gene which a new genome-editing therapy uses to treat sickle cell disease. Her quest for the gene began in another country with another blood disease decades earlier. After completing her Bachelor of Medicine and Bachelor of Surgery degrees at the University of Malaya in Malaysia, Swee Lay went to the UK for postgraduate training in internal medicine and hematology. In 1982, she went to Oxford University to research hemoglobin disorders.
I joined the Weatherall Institute of Molecular Medicine to work with Sir David Weatherall. And he kind of said, well, this is going to be your project. Find out why some patients with beta thalassemia have such a mild disease compared to the majority.
Beta thalassemia is an inherited genetic disease characterized by low levels of hemoglobin and severe anemia, and tissues and organs don't get the oxygen they need to work properly. Patients need regular blood transfusions. Fetal hemoglobin is found in fetuses and newborns. About 3 to 6 months after birth, a switch occurs in everyone where the production of fetal hemoglobin decreases and production of adult hemoglobin increases. Adult hemoglobin is produced by the beta hemoglobin gene. Mutations in the beta globin gene cause sickle cell disease and beta thalassemia.
For a long time, the clinicians have known that if patients with sickle cell disease and beta thalassemia can produce high levels of fetal hemoglobin, they have a much milder disease. But it was difficult to figure out why.
Then Swee Lay hunted for the gene regulating the fetal-to-adult hemoglobin switch. First, she collected blood samples from families with beta thalassemia, including an Indian family.
This spans 7 generations. It even involved a trip out to Malawi to complete as much as possible. So at that time, I could do what we call association analysis within this family. And we found this gene on chromosome 6q.
She found a gene on chromosome 6q which increased fetal hemoglobin and caused mild beta thalassemia in members of the Indian family. But 6q didn't explain the increase in fetal hemoglobin in all families. So Swee Lay hunted for more genes.
People were skeptical that I could actually find this, even including me.
Then Swee Lay collaborated with the director of a twin research unit, and she measured the fetal hemoglobin levels of identical and non-identical twins.
It was so clear that the fetal hemoglobin levels in identical twins were very similar, almost 90%, which then tells me there's a huge genetic component. So I was, felt I can find it.
That twin study provided evidence that she was on the right path and would be able to find the gene that increases production of fetal hemoglobin. Swee Lay had collected blood samples from 5,000 individuals. And she worked with the biostatistician to perform a genome-wide association examination of individuals displaying extreme differences in sickle cell and beta thalassemia traits.
So we took those with fetal hemoglobin levels at the lower 5% and those with levels at a higher, 95% or higher. And we did a scan, a genome scan, using, I think, 300 markers at that time.
Swee Lay found 3 genes that affects the production of fetal hemoglobin. Beta globin, which was known already, the gene on chromosome 6q, which Swee Lay found in the Indian family, and a brand new gene, BCL11A. In 2007, Swee Lay published a paper that concluded the BCL11A gene on chromosome 2p15 is a major regulator of fetal hemoglobin production. Subsequent research by the Shaw Prize Life Science and Medicine co-winner Stuart Orkin discovered that BCL11A repressed the production of fetal hemoglobin.
And this is the area now that is being targeted in gene editing for disabling BCL11A so that it can enable the fetal hemoglobin production to continue in adult life.
The gene therapy stops BCL11A from repressing the production of fetal hemoglobin, so fetal hemoglobin can be produced. The normal fetal hemoglobin compensates for the mutations in the adult hemoglobin, which cause sickle cell disease and beta thalassemia. The therapy can potentially cure people with those blood diseases. In December 2023, the U.S. Food and Drug Administration approved the first CRISPR gene editing treatment for sickle cell disease and beta thalassemia, based on the groundwork of Swee Lay Thein's and Stuart Orkin's findings.
It is a landmark in molecular medicine.
In 2015, Swee Lay relocated to the United States and joined the NIH as the first chief of the newly established Sickle Cell Branch at the National Heart, Lung, and Blood Institute.
Morning, Rick.
The scientific director of the institute says Swee Lay is a special type of scientist.
She's really one of a dying breed of physician scientists, somebody that is an exceptional clinician, truly cares about patients. She's made many important discoveries that have led to new, important, and helpful therapeutics.
William Eaton collaborates with Swee Lay on improving the lives of patients with sickle cell disease.
She has this passion for research and this passion for helping sickle cell patients that is an inspiration to me and to everyone who works with her.
Kong works in Swee Lay's lab. She's taught him to be a better scientist.
As a scientist, she is curious and open-minded, energetic, and also she possesses creative thinking. And as a mentor, she is exemplary and visionary.
Well, Swee Lay is excited that BCL11A is used in a new genome editing therapy for sickle cell disease. Many of her patients won't qualify for the treatment because they have too much organ damage. So she continues to search for oral drugs that will alleviate the suffering of her patients.
One of the things that the patients always tell me is that when they get a pain, they know when a certain pain is going to blow up into a full crisis. So I'm still trying to find a medication that they can take to stop this in its tracks before it blows up.
When Swee Lay is not at work, she likes to take walks around her Washington, D.C. neighborhood to pick up free books to read from a book box.
The color of air. So, nothing of interest today. Oh, I love reading. I mean, you know, I'm a bit of a hoarder of the books now.
Swee Lay's love of books and learning began at a young age. She was born and raised in Malaysia, the 7th child in a family of 9 children.
She paid a lot of attention to study, education. She's the smartest in the family during that time, and she is very focused.
And her advice for the next generation of scientists?
To be prepared to keep learning all your life, actually. I mean, keep an open mindset. Sometimes it helps you think outside the box.
We'll meet Stuart Orkin, the co-winner of the Life Science and Medicine Award in Boston, Massachusetts. The Shaw Prize Special 2024 returns after the break. Stay with us. We're in Boston, Massachusetts to meet with one of the co-winners of the Life Science and Medicine Award. Stuart Orkin is receiving the Shaw Prize for creating a revolutionary gene editing therapy for sickle cell disease and beta thalassemia, which are devastating blood diseases that affect millions of people worldwide. Born and raised in New York, Stuart Orkin attended the Massachusetts Institute of Technology for a bachelor's in science degree. A course sparked his interest in the emerging field of molecular biology.
There were problems to solve, and you know, how does a cell work? How do you express a gene? How do you make a protein? All these things at the time were almost completely unknown. One could feel that you're in sort of an exciting area where there would be discoveries, breakthroughs.
His studies led him to Harvard Medical School for a degree in medicine. After training at the Boston Children's Hospital and the National Institutes of Health, Stuart returned to the Boston Children's Hospital and eventually set up his own lab with a joint appointment at the Harvard Medical School. He always had an interest in studying red blood cells.
At the time I began, that was an area that was sort of the testing ground for molecular biology.
He chose to work on a problem which could potentially help millions of people. Fetal hemoglobin, a protein in red blood cells, is found in fetuses and newborns. About 3 to 6 months after birth, a switch occurs in everyone, where the production of fetal hemoglobin decreases and production of adult hemoglobin increases. Adult hemoglobin is produced by the beta hemoglobin gene. Mutations in the beta globin gene cause sickle cell disease and beta thalassemia. Individuals get sickle cell disease from inheriting one mutated gene from each of their parents.
Sickle cell disease is a disease that affects the red blood cell, and the red blood cell is a round cell, as shown here, carries hemoglobin, which brings oxygen to all tissues of the body. As they begin to sickle, they plug up these small vessels, and then when they're fully sickled, they actually block the vessels, and this leads to tissue damage and pain, the pain crisis of sickle cell disease. Pain crises, which are debilitating and often prevent these patients, when they become adults, prevents them from working, prevents them from having, a normal life, and disrupts the whole family, environment.
Beta thalassemia is an inherited genetic disease characterized by low levels of beta hemoglobin or no beta hemoglobin and severe anemia, and tissues and organs don't get the oxygen they need to work properly. Patients need regular blood transfusions.
Blood transfusions eventually lead to what's called iron overload, because there's iron in each transfusion you receive, and that causes damage to many different organs of the body, including the heart.
The key to finding a therapy for beta thalassemia and sickle cell disease was in the fetal-to-adult hemoglobin switch.
It was known from clinical investigation, family studies, that if one has more fetal hemoglobin than normal as an adult, if you also had sickle cell anemia or beta thalassemia, and you had less severe disease.
He and his team hunted for the gene that caused the fetal-to-adult hemoglobin switch.
We had a couple of theories to test, a couple of hypotheses, and over the next couple of years we tested those and they failed. In other words, we had been unable to learn anything about how fetal hemoglobin was regulated.
Then a breakthrough came in 2007 when a group of researchers led by Antonio Cao used a genome-wide association study and discovered that the gene BCL11A was related to fetal hemoglobin, affecting people in Sardinia. Stuart's lab helped confirm the result. Months earlier, Shaw Prize Life Science and Medicine co-winner Swee Lay Thein had published a paper showing that the BCL11A gene on chromosome 2p15 was associated with fetal hemoglobin. Stuart dived deeper into the relationship between BCL11A and fetal hemoglobin.
When we did that experiment, when we reduced the level of BCL11A, the level of fetal hemoglobin went up, which suggested that BCL11A was a repressor or a brake on fetal hemoglobin.
In another crucial experiment, his team took away BCL11A from the red cells of mice engineered with sickle cell disease.
And what we found was if we took BCL11A away, sickle cell disease was vanished.
Next, he used a novel genome editing technology called CRISPR to edit BCL11A.
The CRISPR comes in, and one can think of it as scissors or tweezers that cut the DNA at a particular place. The DNA is then repaired, and the BCL11A gene is now modified. It's expressed at a lower level, which then turns back fetal hemoglobin production on and is beneficial in both sickle cell disease and beta thalassemia.
The gene therapy stops BCL11A from repressing the production of fetal hemoglobin, so fetal hemoglobin can be produced. The increased fetal hemoglobin compensates for the mutations in the adult hemoglobin, which cause sickle cell disease and beta thalassemia. The therapy can potentially cure people with those blood diseases. And in December 2023, the U.S. Food and Drug Administration approved the first CRISPR gene editing treatment for sickle cell disease and beta thalassemia, based on the groundwork of Shaw Prize laureates Stuart Orkin and Swee Lay Thein's findings.
It's enormously gratifying to see this come to an approved therapy.
The first approved therapy using CRISPR was a momentous achievement in science.
So it's very encouraging for a lot of other scientists in the field. And they have confidence that CRISPR can succeed in an actual patient. So not only is the impact for sickle cell disease and beta thalassemia patients so important, but also as an inspiration for other disease therapies. We know that CRISPR can work in people.
Yeah, he'll do fine.
Daniel trained in Stuart's lab when many of the pivotal discoveries related to BCL11A were made.
He's just like, in a way, the preeminent hematologist of the last generation who's made a huge contribution to our understanding of the blood system.
Malu is from Shandong Province, China. He enjoys working in Stuart's lab.
He's a very nice person and give the postdoctor freedom to pursue their interest, and you can design your experiment by yourself, and it's very important for research.
David Nathan used to lead the Boston Children's Hospital's hematology unit. He saw the potential in Stuart when he was an intern in the 1970s and convinced him to join the hospital.
He was so good that I truncated his fellowship completely. He did very little clinical training. He went right into the laboratory in about 3 or 4 months. And he's been in the laboratory ever since. And he's made one magnificent contribution after another. I'm very proud of him.
Stuart is thrilled that a genetic therapy for sickle cell disease and beta thalassemia has been created based on his research. But the therapy is expensive and complicated. He is working on developing drugs to turn fetal hemoglobin back on, which is a therapy that would be more accessible to the millions of people with sickle cell disease and beta thalassemia around the world.
That if one understands the mechanism and understands the protein involved, that is BCL11A, we can, with modern technology, develop small molecules that will become drugs eventually.
Outside of the office, science is still a big part of Stuart's life.
I think they're downstairs.
His wife is a fellow scientist, and his daughter is a nurse.
His love of learning influenced his parenting because that was kind of like the #1 value in our home was education and learning.
His family is very proud that he's won the Shaw Prize for his discoveries in treating sickle cell disease and beta thalassemia.
It is one of the wonderful ways to show that his peers recognize the importance of the work that he's really spent his life doing. So we're delighted.
While a career in scientific research is often filled with failure and only occasional success, Stuart advises young scientists to be patient and persistent.
Many of us are problem solvers. We want to take a problem, even any problem, and figure out the answer. I want to do the crossword puzzle, figure out the answer. I think it's much the same. We're excited by discovery, finding out new things, and then seeing what that brings as the next opportunity.
Join us next week for more of the Shaw Prize Special 2024 and meet the winners of the Mathematical Sciences and Astronomy Awards, plus an Indian laureate.